Okay, got it. The blue one is actually CA, orange one is actually leukoplakia, whereas the gray one is actually healthy controls. Here also you can see that the leukoplakic samples as well as the cancer samples, they tend to segregate, which means that the dysbiosis that occurs in oral cancer is happening at a pretty earlier stage in leukoplakia as well. Whereas you can see here, healthy controls is staying away. So the group consortium is totally different in healthy controls, whereas you can see the cancer and the leukoplakia is quite tend to segregate together. So we found 14 discriminant taxa which could help in differentiating the healthy controls as well as cancer, but there wasn't much difference between leukoplakia and cancer. So what we concluded was that this microbial dysbiosis, which is seen in cancer, is happening at a very early stage, because that's the reason we couldn't identify much differences between leukoplakia and cancer, but we could see a major differences between control and cancer. Let's see, where is my slide? Moving. Okay, so you can see we looked into the specific taxa where we found that top 10 discriminative taxa, there was difference between these. So you can see there is Porphyromonas, Fusobacterium, Salmonella, Prevotella, again another species of Fusobacterium, Megasphaera, and Streptococcus, which is totally different between these three groups. Now this, again, as I mentioned, we showed that there was a lively bacterial shift in leukoplakia, which actually resembled the cancer bacterium. Now, since we found this in both of this, then we started looking at whether there is a similar change in smokers, whether this change is due to the smoking or the tobacco chewing nature of the people rather than just, looking into just between cancer and controls. So we looked into another cohort, which is like a smokers and tobacco chewers along with normal controls, whether there was any changes. And my slide not moving. Yeah, and there we found that again there was difference, and this changes in the Fusobacterium, what I was focusing, happened at a very initial level. So you can see these changes are seen in smokers as well as chewers when compared to the relative control. So what we found that we found an elevated amount of Fusobacterium saccharibacteria in smokers as well as Fusobacterium campylobacters in chewers. So from there we concluded that this smoking or chewing actually predisposes that environment. We all know that smoking predisposes to an anaerobic environment, whereas chewing also predisposes to an anaerobic environment where these bacteria are enriched already in the process. So these are not cross-sectional studies. We cannot actually tell whether what is happening at what stage, but these are all cross-sectional data. So these are the major limitation of the studies happened until now, like our group as well as other groups. They are all looking at cross-sectional studies to really see where is this microbial dysbiosis, how clearly it's affecting the cancer. We have to look into actually longitudinal data. Okay, so this is the paper. Again, so basically what were the conclusions were microbial dysbiosis is happening in oral cancer and pre-cancer. But we are still at the association stage where we only see these are, we can say this is associated, we cannot say this is causing. To be frank, after decades of research in oral cancer regarding whether it's genotics or whether it's genomics or epigenetics or immunology, we still are at the association level. We do not say, okay, if this gene is altered, you will get cancer, right? We do not have an answer like that until now for the oral cancer. So that's the reason why my role of microbiome is also generating a lot of interest. Now, what the future people are trying to look into is this, how this microbe is interacting with the genes. So there should be some interactions going on there. But as of now, I can say we still have data on association, not causation. But still we have some clues because we have certain bacteria to target, just like colorectal people have actually classified Fusobacterium into the rank of Oncobacterium. Not only my data, we have a lot of studies where we have around 20 to 25 studies conducted throughout the world, in some parts of China, US, some parts Middle East. So most of them, the conclusion is that periodontal pathogens are to target. We have Fusobacterium, P. gingivalis, we have Prevotella. So these are the major pathogens to target if we are actually coming back and looking into the traditional method, that is the culture-dependent methods. And obviously moving further, rather than who is there, as I mentioned, oral microbiome research is still at a very niche initial level. We should look into what are their functions, what are they doing there, rather than just who are they. So recently we did 2 reviews. So this reviews also where we looked into all other studies as well. There we found that most of this data is actually concluding at this, that we have a dysbiosis in oral cancer. The association to causation is still a long way to go. The periodontal pathogens are the ones to target, and we also have the functions to target. I've just summarized these data where they have done. So as you can see, most of the studies. So you can see the studies mostly are from 16 to 17, which is quite recent. We have one study quite early. So it's like in this 2016 to 17 is the time that generated a lot of interest in this microbiome and cancer. So I think we have around 25 studies when we were publishing this in 2020. So recently, maybe more than that, we have around 30 and more than that studies where they are trying to explore. So these are some other of the papers. Again, you can see most of the studies are mainly from US and Taiwan and China region. Yeah. So that's the conclusion from these studies. Okay, so again, moving back, the role of bacteria in oral carcinogenesis, as I mentioned, our studies as well as previous studies have shown that they are actually playing a role in all the 8 hallmarks of carcinogenesis. If you can see here, they have individual roles in proliferation, but how they are working as a group, we have yet to explore. So coming to future perspective, so I just showed you and discussed with you what is the current status of oral microbiome research in oral cancer and pre-cancer. So the future perspectives would be moving on to metabolomics and metagenomics. So metabolome means studying the function, right? Metabolism or whatever they are working. So this is again an omics technology where we can study the metabolism of whole bacteria. I showed you in some of our papers, we showed that the functions is totally different. So same way we have to move on further, looking into the whole metabolome of these bacteria. And another method would be metagenomic and metaproteomics, where we study what are the proteins involved. And there is also some technology called as metatranscriptomics, in which we will look into what are the proteins they are expressing over there. So everything is this meta and omics at the end, but what we are looking is basically the overall picture. Another area to work on would be, though our study is one of the pioneer studies which shows there is a difference in bacteria in the saliva. We were not able to target 1 or 2 or 3 bacteria. There are other groups as well. There are a few groups in New Zealand and USA which are also working on this. We are trying to use this microbiome to develop a diagnostic or a prognostic tool for the oral cancer. So that's another field where people are working on. And another field would be animal studies. So if you, I think I was expecting that question, but anyway, I would say most of these data are from clinical studies. If you can say 1 or 2 people have actually looked into animal studies where they have looked into whether this introduction of Fusobacterium or any of these P. gingivalis can actually increase the size of a tumor or can initiate the development of tumor in mice. So recent papers, I think in 21 and 22, 1 or 2 papers in Journal of Oral Microbiology. So they have mentioned that it can, this means if you are actually introducing Fusobacterium, cannot initiate actually, but can increase the size of the tumor in mice. So we have 1 or 2 data, but yet to be validated. With 1 or 2 data, we can say there are more groups. So that is also a future perspective of if any of you want to work on, where you will look into these role of bacteria in a mice model. But studying microbiome, we have to be a little bit careful, which I will be speaking about in the last section, because it's a bit technique sensitive. It's not like a normal studying a normal gene or something. And finally, again, we go back to a traditional method, individual pathogens. We can still look into the role of these bacteria, because what are the interaction between these bacteria as well as those genes which are involved already, shown to have some role in the development of cancer. We have a multitude of genes that has been shown to have some role. Again, not individually, but in a group, there are a lot of genes involved. So there is this, we can always look into what are the roles of these bacteria and what are the interaction between these roles, whether these bacteria increase the expression of these cancer, decrease the expression of these genes, etc. So these are the 4 pathways. So I just showed you why we have the current focus is on that Fusobacterium. I spoke about the Nature paper. This is a Nature paper, which is currently cited multiple times, because we have got something other than H. pylori to focus on as an oncobacterium. Okay, so as I mentioned to you, some of our recent works basically is we are also looking into whether the Fusobacterium can have some association with genes. So CCL2, I just show you an example where we found that there is a plausible association between Fusobacterium nucleatum as well as CCL20, which is one of the, again, implicated in the development of cancer. So this is a very vast field. As you all know that there is thousands of genes which has been implicated in individual studies in the role of development of cancer. We can always link them. I see that there might be a lot of future postgraduates, not future postgraduates, with postgraduate students here. So that's why I'm showing some of the future work that we can focus on here. And we are also trying to develop a microfluidic system, or what I would say, whether we can use microfluidics using saliva, where we can focus on 1 or 2 or 3 bacteria, which can be developed as a diagnostic or a prognostic tool. Just some of the initial work, nothing as of to show as data. So coming to the last part of my talk, points to ponder in microbiome research. So I showed you what is the importance of microbiome, what have been done in oral cancer and pre-cancer. Now this research is considered to be a bit different from what we do regarding when we are studying genes or proteins or stuff in oral cancer research. So the first thing would be study design, because when we are actually doing study design, the inclusion and exclusion criteria tends to be important. Now, this again is a criticism to our own studies, because I'm sure I saw some of the senior faculty here, so I'm sure they're going to ask, how about periodontitis? There is always a link between periodontitis and oral cancer. And oral cancer is always seen in elderly, and periodontitis is always seen in elderly. So we also have a recent paper which says periodontitis, people with periodontitis have almost 3 point something odds of developing an oral cancer. Again, the common factor is chronic inflammation. So we cannot exclude, we cannot find oral cancer patients, those who do not have periodontitis. So it's a bit difficult, but when we are actually taking the sample, it has to be in a way that, you have to take the sample with all the aseptic precautions involved. So when you are taking the sample collection, you have to make sure that the area is very aseptic. So whenever we are collecting the sample, we clean that area with betadine, then we take the bacteria whole sample, and then we section it, and we try to take it from interiors of the tumors with as much aseptic precautions. And we do it in special chambers, BSL2 labs, where we are trying to make this thing as aseptic as possible. So aseptic precautions is something that we have to do in microbiome research. Anything can contaminate the microbiome research. You take a sample, you're transferring the sample. We have got samples. When we sequence it and when we generate the data, there is only E. coli. So E. coli is a potential confounder also. If we can clearly understand, if we find a lot of environmental contaminants, that means our data is useless. So that is something that we have to be careful about. So inclusion and exclusion criteria, something to focus on. Sample collection. aseptic precaution, sample storage. So has this microbe has to be stored immediately in frozen conditions, because if you are keeping that sample for long, some of the pathogens may multiply, whereas some of the pathogens may degrade. So again, that alters your data. So immediately after the sample collection, you have to store them. And then all the transport has to be on ice and has to be stored immediately, freeze them immediately, or extract the DNA immediately. Any amount of storage in normal room temperature or even 4 degrees is actually altered the data, right? It's completely alters the data. Sometimes E. coli might just increase because, you know, our precautions, if we have not taken any precautions. Again, there is sample processing, sequence assembly, and also, you might have thinking, how do we generate these data? So basically, once we have all these ATCG or these data, we are actually blasting them to a certain amount of databases. So there are these databases, we have PubMed databases, we have private databases. So whenever these databases may also have a role in this changing the results, because a little bit, because when we are actually trying to generate what are the data, depending on the databases, there might be few differences. So there is a need to be standardized, and also the downstream analysis and comparison. So one of the limitations that I found in this microbiome research is, like, you know, though there are 30 studies, different people have done different comparisons. So maybe that's the reason we were not able to find 2 or 3 bacteria or a consortium which we can actually target on. So as of now, we just got Fusobacterium. Some studies have given Pigeon-Givalis, whereas few studies have given Prevotella. But I cannot say Fusobacterium was shown to be upregulated in all the 30 studies. So there could be some I would say, implication of all these study design, sample collection, sample storage, and as well as these downstream data analysis on the results. But still, though there are so much of differences, we still got similarities with some of these bacteria, regardless of where these studies were conducted. So if I've shown you studies from China, shown you studies from US, Taiwan, New Zealand, India, but still we have some of the differences. So that means, some of the similarities, which means that there is an amount of microbial dysbiosis, and the microbial dysbiosis is something to consider in the future research whenever we are planning anything, any kind of research, as well as, I would say, any kind of treatment planning as well in microbiome research. So if some of you are interested in understanding the methodology, I have a chapter in this book, where we are actually delineating all the specific precautions we have to take when we are doing study design, sample collection, storage, sample processing, etc. So takeaway messages. So I would say there is a definite amount of microbial dysbiosis that is occurring in oral cancer and pre-cancer. And association into causation is a long way to go. And I'm not sure whether we will be able to point out one bacteria. We wouldn't be, right? We have been studying oral cancer for such a long time. And we know that tobacco is one of the initiating factors. But still, with all these data that has been generated, if you're working on this, obviously we can find out there should be some role in progression of oral cancer, and why somebody is getting an oral cancer, but why somebody is not getting, despite they have a common etiology of use of tobacco. So this is a very advancing field of cutting edge research. If you see this genome project as well as microbiome project is hugely funded in most of the countries, whereas in India, we are suddenly just trying to start it off. Gut microbiome, obviously, yes, we have data. Oral microbiome still at a very nascent or initial stage. And it's also time to move on. What the world is now focusing on, this is all, though, so there is one. So I was fortunate enough to work with one of our very senior faculty or senior researcher, honorary researcher, that is Prof. Newell Johnson. So one of the things that I have learned from him, though whole of the literature we are saying microbiome, he is very particular that we should use only bacteriome here, because we are only focusing, not only us, the whole field as of now is only focusing on bacteria. the published studies. So that's why we tend to use the term bacteriome. But if you look into other papers, they have used the term microbiome. So as of now, the data, to be correct and precise, it is only bacterial data, bacteriome. But we also know that there are many uncultivable viruses as well as funguses. So that's why the future would be to look into virome as well as microbiome. Only these three will constitute microbiome. But this is just our way of putting it. But if you just look into literature, just Google it around, you see bacterial papers already classifying or publishing as microbiome. So as I mentioned again, metagenomic, metatranscriptomic, metaproteomic, and metabolomic approaches are the future. But depending upon the funding, we can also look into single bacteria and its association with single gene, because another limitation of microbiome funding is its cost. Because since this is cutting edge and next generation sequencing, the funding has to be huge. And finally, we also have to look into animal studies, where we can see if there is an association, we can see whether there is any association in vivo between the development. There are 2 or 3 papers. If anybody is interested, you can always email me. I can send you. So that shows that Fusobacterium has been shown to increase the size of tumors, but nothing as such shows that it initiates the cancer. It improves the size of the cancer. Okay, so that's all from me today. I hope I was able to generate a little bit of interest in this very new field. So once when Professor Sunil actually approached me, I was thinking, rather than speaking about oral pathology, where we have very senior people to do and address rather than me, I would introduce to you with this very new field, which I have been fortunate to work on for the past years, so that I would be able to spark some interest to look into this oral microbiology as a part of oral pathologist, because if you really know, our degree is actually oral pathology and oral microbiology, not specifically oral pathology. So maybe looking a little bit into integrating this microbial aspect into oral pathology as well. So that's all from me today. I'd be very happy to share and for your questions, as well as I would prefer we can do a discussions. I see Babu Matthew sir here and many of my senior teachers. So I'd be happy to have a good discussion. Thank you so much.
Thank you, Dr. Divya Gobinath, for the astonishing presentation. Respected chairpersons, now the session is open for discussion.
Thank you. Thank you, Divya, for the highly informative topic. And also, I got about regarding the dysbiosis which takes place in periodontal disease and in caries, and its association with potentially malignant diseases as well as oral cancer. Is there any association with the dysbiosis related with periodontitis as well as dental caries?
Sir, it was a bit not clear, sir. I mean, there was a disturbance. Sir, can you?
Hello, can you hear me now?
Yes, but a bit of disturbance. Yes, sir.
Hello.
Yes.
Again, I'll repeat the question. The dysbiosis which takes place between periodontal disease as well as dental caries.
Oh, yes. So this data, if you see, so I was just focusing on oral cancer and pre-cancer. So there are a lot of studies, actually, this oral microbiome, they're also looking into caries and periodontal diseases as well. So periodontal diseases, as we all know, we still do not have one pathogen to target for. We have these complexes. So periodontal diseases, I would say one of the initiating diseases where this whole concept of microbial dysbiosis actually came from. Right? We also have learned that it's even with the culture-dependent studies, we know that it has always been a consortium rather than single bacteria. So there has been studies going on, and So, but still, as these bacteria has already been proven as one of the initiating factors, the studies are not able to conclude anything further. So we have all these sets of pivotal, all these complexes. So they are trying, and so what the studies have proven is what we already know about it. So it's a consortium or a series of bacteria that's happening in periodontitis. Regarding caries, as I mentioned, yes, nothing further I would say. Still, they are revolving around streptococcus and different species of streptococcus as an initiative factor in caries. So yes, so apart from these two, what other area these oral microbiome they're looking into is systemic diseases, especially autism, all these major kind of depression, central nervous diseases, so the central nervous system diseases, gastric diseases, gastroenteritis. So now we have found that there is a microbial dysbiosis in majority of these systemic diseases that has been already, I would say, major of the systemic diseases. So that's the area where most of them are focusing on rather than caries and periodontitis, because it has already been proven. Did I answer your question, sir?
Sir, please unmute, sir.
Hello, can I speak now? Hello, I would like to thank Dr. Gopinath for an excellent presentation. As she has rightly put out, from the very inception, the degree in oral pathology is oral pathology and bacteriology. But there is hardly any work being done by the oral pathologists in the area of microbiology. She has emphasized this is time that we concentrate in the microbiology area also. I have to make one observation. Ethanol was considered not as carcinogen until very recently. It's been listed as carcinogen because in the oral cancer, alcohol gets put into butyraldehyde by certain organisms, and that's why the oral microbiota, which is capable of converting ethanol to butyraldehyde, is important in the causation of cancer. Another thing is that in case with the superadded candida infection, where a higher rate of malignant once again congratulate the organizers for having included this new area of knowledge in the seminar. Thank you all. That's all what I have to give as comment.
Thank you, sir. May I just add on what sir just mentioned? So as sir mentioned regarding that ethanol degradation, right? One of the reasons, so in our data, sir, I showed you that the metabolome of the oral bacteria in oral cancer patients had increase in that ethanol degradation. That is one of the, Neisseria is one of the pathogens who do this ethanol degradation. So that was increased, that ethanol degradation pathway was increased in oral cancer patients in our cohort. So as sir rightfully mentioned, so these are the area that, there are some hints saying that this really, they do some work over there. Thank you, sir. Yeah, for pointing out that one.
Over to you, respected chairpersons.
Thank you, Divya, for that excellent and informative session. And I'm like wonderstruck by the epic proportion of data you have amazingly collected and you have published. Hats off to you, Divya. And I'm proud to be associated with you from long back, and the data is amazing. And thank you for that excellent presentation. And thank you also for reminding us about the importance of microbiology. Usually our degree is oral pathology and microbiology, and that microbiology part is often forgotten. And thank you for bringing it alive. Thank you, Divya.
Thank you, ma'am. Thank you.
Dr. Divya Govidath, kindly receive the certificate as a token of appreciation.
Thank you. Thank you so much. Yeah, thank you so much.
Dr. Padma Umar sir, kindly receive the certificate as a token of our gratitude.
Okay, thank you, Sherlyn.
Dr. Sujata Verma ma'am, kindly receive the certificate as a token of our gratitude.
Okay, thank you, Sherlyn.
Dr. Padma Umar sir, kindly receive the certificate as a token of our gratitude.
Okay, thank you, Sherlyn.
Dr. Sujata Verma ma'am, kindly receive the certificate as a token of our gratitude.